Good afternoon. Welcome to ASCP's Best Practices in Advanced NSCLC Project Echo video conference series. Today's session focuses on advances in targeted therapies for advanced NSCLC. My name is Kelly Beimer. I am ASCP's Director of Learning Innovations, and I'll be the facilitator for today's session. A copy of the disclosures can be seen on the screen. And in addition, today's session is funded by independent educational grants from Janssen Biotech, Inc., administered by Janssen Scientific Affairs, LLC, and by Pfizer, Inc. Pleasure to welcome Dr. Christian Kunder to today's Project Echo video conference. Dr. Kunder is a clinical associate professor in pathology at Stanford University. He also practices at Stanford Hospital and Clinics, as well as the Lucile Packard Children's Hospital. He'll be sharing his expertise on advances in targeted therapies for patients with advanced NSCLC. So now I'll turn it over to Dr. Kunder to share his slides and begin.
I am going to try to give a brief overview of this large and very interesting topic. It's something that we're all thinking about a lot, and it's a moving target as well, so it's a lot of work. Just for a little bit of historical perspective, this story really begins in the early 2000s with this failed clinical study, Fitinib, which was one of the first small molecule tyrosine kinase inhibitors for EGFR. And there was no improved efficacy over gemcitabine, just a standard chemotherapy and cisplatin alone. But there were clearly patients who were responding, even though percentage-wise it was not better than the chemo. At about the same time, we were discovering that there were highly recurrent mutations in EGFR, the epidermal growth factor receptor, in lung adenocarcinomas. And as you can see, there's several very common ones. L858R and exon 19 deletions up here are both amenable to treatment with the first generation EGFR inhibitors. So eventually it became clear that it was the patients with the mutations that were responding. And then shortly after that, we discovered these exon 20 insertions in HER2 in the kinase domain, which is still a bit of a problem actually in terms of targeted therapy. But ALK came a couple of years later, ALK fusions, and basically the story has been we've been identifying smaller and smaller slices of the pie as we go. But these discoveries came along with the pretty rapid development of small molecule inhibitors, as tyrosine kinases are somewhat amenable to that compared to other protein targets. And so we got a lot of studies like this showing that in this waterfall plot, for example, that there were extremely dramatic responses in patients with ALK fusions to crizotinib, and most patients with ALK fusions had some response at least, if not a major response. This is the same type of study using crizotinib in ROS1 rearranged lung cancer. Again, almost everybody responds, at least for a while. And so these drugs were pretty quickly approved by the FDA, since these responses are significantly better than what you would expect from standard chemotherapy, which was the standard of care at the time. More recently, we've discovered these exon 14 skipping mutations in MET, which is an interesting alteration and a little bit hard to identify. So it wasn't clearly an activating mutation at first. But again, we have the trial here with capmatinib, which is now FDA approved for patients with MET exon 14 skipping mutations, showing very dramatic responses, even in previously treated patients. Basically, each one of these driver mutations in lung cancer gets its own New England Journal paper, and that's probably very nice for the authors. Again, this is just a reiteration. This is the most recent one, selpercatinib in RET fusion positive non-small cell lung cancer. And I'll just talk briefly about the MAP kinase pathway, which I'm sure you're basically familiar with, but essentially you have a receptor tyrosine kinase in the cell membrane, and these are examples of the ones that are most relevant to lung cancer. And ligand binding ordinarily causes these to dimerize and phosphorylate each other, which sends this the rest of this pathway into action, activating RAS, RAF, and then the MAP kinases, of which there's a bunch. And so you can see that of our driver mutations, this is some data from Stanford in terms of just percentages of lung adenocarcinomas with driver mutations. You can see that we're still getting, we still don't know in 19% of cases, and that's been a pretty durable number for a few years now. But the proportion of patients that have alterations that are now targetable is increasing over time. So in our population, we have mostly EGFR mutant lung cancer, which is generally associated with non-smokers, and somewhat less KRAS-driven lung adenocarcinoma. These KRAS-driven lung cancers are more likely to occur in smokers, and so the percentage of those decreases as people stop smoking. But of course, you can get either one in either situation. The other pieces, as you can see, we get down to 4% very quickly at the MET exon 14 mutations, ALK fusions, HER2 mutations, which actually there's not an FDA-approved drug for yet, although there's some off-label uses. BRAF, which I don't think I mentioned, but a fair number of lung cancer patients have BRAF V600E and do respond to combined therapy with BRAF-directed therapy and MAP kinase-directed pathway. So then once we get down into these fusions, the groups are smaller and smaller. And you can see that in our 900-something cases, we only saw like one case of an NTRK3 fusion and one case of an NTRK1 fusion. And I'll get into this a little bit more later, but partly that is an assay limitation. And that's something that you have to be aware of as a person ordering these tests, is that the negative predictive value for each kind of alteration is not equal in general, even if the genes are covered by the panel. For example, we only got lucky to detect this one NTRK3 fusion because our panel is a DNA-based panel, and we are not covering the enormous intron where most of these rearrangements take place, as it would take up, that would end up being like a half of the panel or something based on its current size. So for these fusions, it's reasonable, and the NCCN guidelines say now that it's reasonable to test those patients with an RNA-based method, which is much more straightforward since the coding sequence of a gene is much, much shorter than the actual entire sequence of the genes. And the breakpoints do tend to happen in those introns, so you get exon from one gene, exon from the next gene. And if they're in frame, then you get an actionable fusion protein, right? There's a couple of other ones where not clear, not super highly agreed upon that these are really drivers in lung cancer, and RAS probably yes. NF1 is also in the RAS pathway, and these mutations are common in all patients with lung cancer, and there's just a few that only have, that's the only thing they have that we're detecting. There are certain other kinds of alterations too, which are being described, which can also be difficult to identify. I think an example is EGFR kinase domain duplications, which require some planned intron coverage ahead of time, and also the bioinformatics tools to call complex variants like that. It's super rare. It's like 0.1%, I think, overall, but that's what the game has been about, is trying to get everybody that is in a potentially treatable category to know that they're in that category. These are the currently approved, FDA-approved drugs for lung cancer, and as you can see, it depends on not just what gene is mutated, but even what mutation you have. These exon 19 deletions and L858 are by far the most common mutations, and they respond to the whole spectrum of small molecule tyrosine kinase inhibitors. These exon 20 insertions, with one exception, generally do not respond to tyrosine kinase inhibitors, but this is within the last year we had these two drugs approved, which actually are effective in this context. It might be effective in the HER2 activating mutations also, but it hasn't really been investigated all the way yet. But these exon 20 insertions in HER2 are sort of paralogous to the ones in EGFR. There are a lot of kind of uncommon genotypes in EGFR, even within EGFR, and there's less known about these, although there is now approval for these three mutations and afatinib. T790M, I'll talk about in a minute, is associated with resistance to the first generation of tyrosine kinase inhibitors. And now we have the second generation, which is able to inhibit T790M EGFR. So these are cases that had exon 19 deletions in L858R, and then their resistance mechanism was to develop this mutation. And it explains about 50% of primary treatment resistance to first generation TKIs. The NCCN guidelines now prefer that you actually start with osimertinib, which is equally effective on the un-T790M mutations. And obviously you don't get the emergence of these clones. As I mentioned, the patients with BRAF V600E can be treated with dabrafenib, which is a BRAF inhibitor, and trametinib, which is a MAP kinase inhibitor. And most of these patients, there are a fair number of patients also with non-V600E mutations, a lot of times as secondary events in their tumors. And these patients probably will not benefit from this kind of treatment yet. It's just they are a very small part of the pie, and it's really these V600 mutations that respond well to current BRAF inhibitors. There are now at least 5 ALK inhibitors, 3 generations, lorlatinib being the first third-generation ALK inhibitor, and crizotinib being the first one. There's not as many options for ROS1. NTRK has been in the news lately, and there are 3 genes here, NTRK 1, 2, and 3, and these fusions are very responsive to this class of drugs, larotrectinib and entrectinib, which were rapidly approved a couple of years ago. And what's interesting about this approval is that it's for any solid tumor with NTRK fusions, whereas almost all FDA approvals prior to that had been based on histology first. So, you know, lung cancer with this or without this. So that was kind of a big step. And it does seem to be quite effective in other tumors with NTRK fusions. NRG1 fusions, this gene encodes neuregulin, which is the ligand for HER2. And there are a few cases where this is highly overexpressed. And this is another thing that we would not catch on our regular panel for similar reasons. And now we have MET-directed drugs for exon 14 skipping mutations or high-level amplification, which is also seen in a certain percentage of cases. The HER2 drugs, as I mentioned, are off-label right now, but just a couple of weeks ago, there was a New England Journal article very similar to the ones I showed you before, looking at trastuzumab deruxtecan, which is a antibody drug conjugate. So the idea here is that the trastuzumab gets the drug to the cancer cells, and then this part, the deruxtecan, is the topoisomerase inhibitor. So it's basically a way of targeting toxic chemotherapy to the cells that are needing to be killed, the cancer cells, and not as much the normal cells. And then, as I mentioned, there are 2 drugs now for RET fusions as well, which are also approved for thyroid cancer. And that seems to be kind of a common trend here, which is that a lot of these fusions especially are seen in other tumors other than lung cancer. And there aren't really FDA approved indications for treating most of those yet, but there does seem to be activity in those cases that have tumors driven by these fusions. So resistance, you know, this is very exciting because patients who are treated with these drugs are not subjected to toxic chemotherapy. And the drugs themselves are not completely benign. It's not something you just want to take to prevent lung cancer, for example, but they are much easier, much better tolerated than chemotherapy is. Patients generally respond to, as you could see in the studies I showed you, but they don't respond forever, and eventually resistance emerges. As I mentioned, in EGFR, T790M mutation was a big problem for a long time. We were testing for this all the time. And then osimertinib was approved in 2017. And in the early papers about this, they predicted that this mutation would cause resistance to osimertinib because osimertinib covalently binds to this amino acid. And sure enough, that's what happens. So now we have some patients who've been treated with first and second generation EGFR inhibitors who have T790M and C797S, and it becomes very difficult to treat those patients. The other basic mechanism for resistance is to turn on another pathway that also activates the RAS/RAF MAP kinase pathway. So MET amplification and HER2 amplification are pretty common mechanisms of resistance. And also in some cases, you'll get a second driving mutation, like an activating KRAS mutation or a different fusion, for example. And these resistance mechanisms are well described for almost all of these that have targeted therapies now. I think all of them, yes. And it is a combination of mutations in the sequence that make the actual protein less likely to interact with the drug, or else activation of a second pathway. And this activation of a second pathway is more problematic because in general, we're getting down to very small groups of patients here. And we don't really have a lot of data on combinations of therapies to try to inactivate different pathways. But a lot of that is probably going to be limited by toxicity, even though these drugs are fairly well tolerated. So as I mentioned, for resistance mutations, you can develop more drugs, and different compounds will interact differently with the target molecules. And that has led to some success, although again, everyone eventually develops resistance, although there can be some very long responses. And I shouldn't say everyone, there are some complete responses as well, but in general, resistance is a problem. There are a couple of studies that have looked at this possibility of inhibiting multiple pathways, and they have shown some efficacy, but it's going to be a difficult area to study. And then, of course, once there are no more targeted options, then we turn to some combination of immunotherapy and chemotherapy, depending on the patient's situation. At which point they are kind of back to where we were in 2004, which is without a lot of time. Aside from immunotherapy, which, as I mentioned, was developed in this period, and these other indications that are tumor types independent, microsatellite instability and tumor mutation burden, can also be used to select patients for immunotherapy, but it's not as common. It's probable that some genotypes also predict resistance to radiotherapy or immunotherapy. And then I would be remiss if I didn't mention squamous cell carcinoma, which accounts for 25% of all non-small lung cancers. The problem here is that you have a very different set of genotypes. Although here you can see that at Stanford, we've tested 66 of these in this one group, and 14% of them had adenocarcinoma-like mutations. And that probably reflects the fact that when you sample a patient's lung cancer and you get a limited sample, you'll sometimes get a squamous area, even if the whole tumor is not squamous. So that's probably what's going on here. And it's also why the NCCN has suggested that broad testing be undertaken for patients with squamous cell carcinoma as well. It doesn't seem like biomarker-driven therapies are really all that effective in this context so far anyway. So, as I mentioned, targeted therapy is a very fast-moving target. Every year I get to add some drugs to the list, and we are still doing most of our testing on biopsy material, although there is also more cell-free DNA being used, especially in the progression situation. And because more of these are actionable than ever, it's more important than ever to recognize the limitations of individual assays, which is a very, very difficult thing to do, really. But it is necessary. All right. So I will stop sharing and mute myself.
Great. Well, thank you so much for such an informative presentation, Dr. Kunder. I would like to open the floor up for a few minutes for any questions or comments from our participants. We would love to hear from you, so feel free to type any questions or challenges you face or any thoughts that you have regarding targeted therapies. So we did have one question come in over the Q&A box asking whether you view MET exon 14 as a complex fusion in addition to being an SNV and indel. And they're also curious about your thoughts on NGS DNA to NGS RNA and the pros and cons of that for NSCLC patients.
Yeah, there's a lot of different variants that can lead to MET exon 14 skipping. And this is one of the assay limitation things I was talking about before, because unless you cover the complete introns surrounding exon 14, you can't ensure that you've detected every possible variant, which is, again, a reason why in patients where you're not getting anything with the initial DNA panel, it's probably reasonable to do RNA-based testing, because there are RNA-based panels that can detect exon skipping as well. It's a challenging thing to detect, I think, because there's probably non-zero exon 14 exons skipping even in the in normal tissue. So you have to be very careful about your thresholds. But yeah.
Great. Dr. Ordulu or Dr. Dong, do either of you have anything to weigh in on the question?
Yeah, I have a question for Dr. Kunder. So my question is, you mentioned many different mechanisms of resistance, and some of which have to do with copy number change. So I was wondering if you report copy number change by NGS, whether you try to quantitate them, differentiate high from low, focal from non-focal, and what are the issues around that?
Yeah, that is a difficult thing. Luckily, most of these resistance-based amplifications are very high-level amplifications, and so it's not really a huge issue. We do report amplifications, and we report them with a minimum copy number estimate, because the other problem is that the more non-tumor tissue you have in your sample, the less impressive your amplifications look, and there's really no way to be sure about what the real tumor content is. Especially given that many lung cancers have genome doubling and all kinds of other stuff going on. So I guess in a way, because there's not really much that can be done based on those findings anyway, it hasn't been a huge issue. But I think, you know, the ideal is that if that was relevant and the threshold was relevant, you'd really have to do FISH, which is a single cell method to count the number of signals.
Well, this has been a great discussion. We're going to pause now and begin our next presentation. So it's my pleasure to welcome Dr. Sarah Ordulu to today's Project Echo video conference. Dr. Ordulu is an assistant professor at the University of Florida, and she's going to be sharing an interesting case related to targeted therapies for patients with NSCLC. And then again, throughout the presentation, we do encourage all of you to type in your questions and comments along the way. So now I'll turn it over to Dr. Ordulu to share her slides.
Hello, everyone. Thank you very much for having me here today. And just before starting the case, I'm currently an attending at the University of Florida, but the case I'm presenting today is from Massachusetts General Hospital from last year when I was still a fellow at the combined program. And just wanted to point out, it was such a privilege to be a fellow there because I got to see 3 different labs and the 3 different labs approaches to molecular cases. So I had lots of insight into how to approach different types of scenarios with different types of troubleshooting at different labs. So I'm very grateful to my training. So let's get into our case and let's talk about some of the technical aspects of these targetable fusions and how we can get some clinical relevance from our molecular results. So the case I'm presenting today is a 64-year-old never smoker female presented with a non-productive cough, and imaging studies revealed multiple bilateral lung nodules with lymph node positivity, and the cytology was consistent with a lung adenocarcinoma. And our molecular testing at Mass General Hospital showed an ALK-EML4 fusion. Before showing you the test result, I just wanted to briefly mention, I don't know about you, but for me, when you talk about a fusion gene, I always kind of immediately think about a translocation and exchanging parts between two different chromosomes. But fusion gene can happen as a result of any structural rearrangement, including inversions or deletions. And this common ALK-EML4 fusion is actually a result of an inversion on chromosome 2. And even though EML4 is the most common fusion partner, when you look at the breakpoints, you can see that even at RNA level, different exons of EML4 can be involved in the fusion, whereas for ALK, it's usually the exon 20 with the kinase domain. So keeping this in mind, we kind of briefly touched upon how to detect fusions by NGS assays. RNA assays tend to be a little bit easier than DNA because they are skipping those big intronic regions and you are getting your exonic reads coming together. But even then, if your assay is targeting both ends of a translocation, you might miss an unknown partner that you haven't been targeting. So one of the cool sides of the platform they use at Mass General Hospital is the ability to detect an unknown target. So they are using this method called anchored multiplex PCR, where you start with fragments of germline DNA or cDNA you create from your RNA, and you start with some adapter ligation that can attached to any type of sequence. And your first PCR reaction is done with a gene-specific primer on one end, which is the gene that you are targeting. And the other end is just a generic primer that can attach to your adapter. So you only need to target one end of your rearrangement. And then you do a second round of PCR. And again, in this round, you have another gene-specific primer. So at the end, you are only uncohering one end of your rearrangement, and the other end can be anything. And by reading this sequence, you can find the fusion partner. So for example, in the case of an ALK-EML4 rearrangement, by targeting ALK exon 20, you can find all the different exon partners of EML4 and even different gene partners. So, and this is just a IGB screenshot of how the fusion looks when we are analyzing in the lab. And this is basically reads from a single amplicon, and you can see on the right, it's partially aligning well to ALK, so you don't see any mismatches. And half of it, it's like there's all these mismatches, and those are the reads that are coming from EML4. And the same is true for the EML4 region here, where your half is aligning well to the EML4 and the other half is coming from ALK exon 20. So this is basically how you can visualize RNA-based fusion assay in IGV. Going back to our inversion in ALK, I just wanted to point out, obviously, the NGS is not the only option. Actually, ALK is a very good example. You can use more conventional techniques and get the same result. So you don't need necessarily a fancy report, a fancy assay to prove there is a fusion. One thing that I wanted to point out again, because it's an inversion on chromosome 2, even though they are separated by millions of base pairs, when you look under the microscope with a fish probe on them, it might be hard sometimes because it's located in this short arm of chromosome 2, and it might be difficult to interpret sometimes the signals. But the idea is when you have a break apart probe, which is, and for ALK, it has been approved by FDA since 2011 as a method to detect the rearrangements. What you expect is in your wild type state, your two probes are together in the 3' end of your ALK. And they are designed in a way that the breakpoint breaks them apart. So once you have an inversion, those two probes then are becoming away from each other in the genome. So therefore you see the signals being red and green and split apart. And as I was saying, ALK is one of the best examples where you have actually some conventional methods available to just go to them without even doing NGS. And this particular antibody with the D5F3 clone for ALK is again approved by FDA, and it's a binary scoring IHC method where if you have cytoplasmic granular staining, any type of staining is considered positive. And there are even papers talking about how different fish signaling can also correlate with the intensity of your IHC results. For example, this one paper says when you have your regular split apart probe with two split and one fused, you have your strong expression, and then in some scenarios where you have this weird signaling where you have the loss of this fused signal, but you only have the split signal, it has some weak staining. So regardless of this type of pattern-based approach, we just call any staining positive, and it's been shown that there's a high percent concordance between fish and IHC, so IHC can be used as a standalone test, although we know that there is some discrepancies as well. But even for the discrepant ones, it looks like the ones that are ALK IHC positive and fish negative still respond well to treatment, so that's why IHC is a really good method to use in the routine practice. So now going back to our case, we have the ALK EML4 fusion. So the patient just thinking about targeted therapy, usually what we get is now we have basically 3 generations and even more ALK tyrosine kinase inhibitors, and usually historically it has been that you start with crizotinib and go from there depending on the patient's response, and this is what happened to this patient. So the patient got crizotinib therapy after the detection of ALK fusion, and within a year she basically had complete resolution of her tumor. Unfortunately, at 3 years, she came back with additional right lower lobe nodules and pleural effusion, and at the time, the cytology showed again the lung adenocarcinoma. So she was started on the 2nd generation ALK TKI inhibitor alectinib. So within a couple months, she started to show treatment response, but unfortunately, in the same year, she continued to progress on the alectinib treatment, and we got a pleural biopsy at the time. And in this pleural biopsy, in addition to the ALK fusion, now we detected in our tumor SnapShot sequencing, which basically looks at the mutations in the tumor. We found these 2 different mutations. One was a loss of function mutation in ARID1A, and the other one was a ALK mutation that was in the tyrosine kinase domain region of the ALK. And with this, I just wanted to talk about different resistant mechanisms you can get for ALK rearranged tumors that are treated with ALK TKI inhibitors. So basically, I kind of think this is kind of akin to microbes having resistance to different antibiotics. They change their genome and they kind of find another way to invade into the host. And it's kind of the same idea with the tumors with their fusion genes. If you basically, once they get a mutation, they can resist to the treatment effect of the inhibitor. And they can do this directly by getting resistance mutation in the tyrosine kinase domain that could affect the binding of crizotinib and having the act of crizotinib on the ALK fusion itself, or they can have some bypass mechanisms where you have, instead of having the ALK activation, you can have some downstream effects that can result in the same cell proliferation and survival result that ALK would provide. And in our case, we got both an ALK resistance mutation in the tyrosine kinase domain, but also the ARID1A mutation. Even though it's not one of the most common things that has been mentioned, I can foresee this as a downstream pathway alteration because ARID1A is effective on the PI3K pathway. And by inactivating ARID1A, you can basically are activating PI3K pathway. So it's kind of like a bypass mechanism as well. So the patient unfortunately had both type of mechanisms. So within a couple of months, the patient started on another 2nd generation ALK TKI, and unfortunately the lesion stayed stable for a while, but then again she had worsening symptoms and got another pleural biopsy. And at this point, she had 2 different ALK alterations, mutations, and 2 different ARID1A mutations in her tumor. And for this one, I just wanted to point out one thing that we don't necessarily consider with NGS testing is that we just see a list of mutations, but we don't think if it's in the same clone or is in the same allele. So the couple things to consider is that these are coming from all the same cell type or the same cell in the tumor. And even if it's coming from the same cell, is it in the same allele within the tumor? So the only time we can be certain with NGS-based assays like this is when you have two alterations that are in close proximity enough to see on the same read and to tell, okay, they are in cis, which means that they are in the same clone and they are in the same allele. For everything else, it's kind of, I guess, guessing game. You can kind of look at the allelic frequencies and stuff, but you can't necessarily tell for sure that they are all in the same allele or clone. In this setting, because both of the mutations were far apart, there was no way of telling whether or not they are in the same clone or cis or trans. But we could tell that these were different mutations than it was detected on the prior treatment. So this at least tells you that whatever subclonal events were happening at the beginning, this is another clone now is taking over after the second therapy, that now the clone with the different mutations is taking over in this patient. And another technical aspect I wanted to point out is that these are, again, for NGS-based methods. The SnapShot has primers enriched for regions that we are looking for mutations, whereas Fusion has primers that are kind of targeting most commonly altered regions. But technically, if you can enrich your Fusion panel for areas that you know that you have resistance mutations, you can actually find your resistance mutations in your Fusion panel. It's just that our platforms currently are not designed to find mutations in your Fusion panel, but if you go to your raw reads, you can actually try to find your mutations yourself. And in this particular case, I was just curious, can I catch any glimpse of this resistance mutation in the Fusion panel? And there were a couple of reads that were randomly kind of generated, even though they weren't specifically targeted, but you can't obviously make any claims from these reads, but you can see that there's a clue of this resistance mutation in the Fusion panel. And if we were enriched with the primers in this region, I'm pretty sure you could get a better shot of this rearrangement mutation in the Fusion panel as well. So that's something to consider for creating your assays now that we know the most common resistance mutations and everything. Maybe with just one test, you can also test for your mutations in the same Fusion panel. Again, going back to all the different generations of tyrosine kinase inhibitors, now this patient went through crizotinib, alectinib, and brigatinib, and the last resort is usually in the clinical world is the lorlatinib, which is what this patient received. And again, there was some minimal response, but in a couple of months, or in a year, she got additional nodules and a breast lesion. And in the breast lesion, now we have the same mutations we have detected in the prior, and now an additional ALK mutation. Again, unfortunately, because they are so far apart from each other, I can't tell whether they were in cis or not, but at least now we know that the same mutations still exist in this tumor. So it's kind of some sort of subclone of the previously resistant tumor. And now it has a new mutation. And at this point, the patient is now receiving chemotherapy combined with lorlatinib, and they are considering some clinical trials for some advanced therapy. So this is not our case, but I just wanted to point out again this clonal evolution phenomenon in multiple generations of ALK inhibitors. So in this particular paper, they were actually able to track each clone of the tumor and see what type of mutations they got after each therapy. And you can see at the beginning, you have your resistant mutations, just one of them, and they each have their own clonal evolution. And after a couple of ALK TKI therapies, you'll get this bottleneck that your pre-existing ALK mutated tumor gets an additional resistance mutation and then becomes resistant to your final therapy, which is the lorlatinib. So now it's been an increasingly recognized phenomenon that your single mutant active tumors becomes even more resistant with the double mutations in cis. And here's just another example, and this is what I was talking about when I talk about having the amplicon being able to show the mutations in cis. In this particular case, the two mutations the patient got were close enough in the gene that to show they were in cis together. So, and this is another indication that this patient developed resistance to lorlatinib. So this is an increasingly recognized phenomenon now that with the multiple lines of therapy, if you start getting double mutations, you will get resistance to this second or third generation inhibitors. So now the paradigm is basically, instead of first, second generation, should we kind of look at the patient's mutational status and decide on our treatment based on that. And another thing to mention is that crizotinib is known to be not responding well to CNS disease, CNS involvement. So for example, at the beginning, if you already know your patient has a CNS metastasis, do you want to start with your second generation inhibitors? And if you have double mutant disease, now there are this new drugs out there that they are kind of doing clinical trials to inhibit the ALK through this way. Again, going back to this picture, though, we are kind of still only talking about the resistance mutations in the tyrosine kinase domain and not necessarily even touched on the bypass mechanisms. And when you look at their percentages in overall resistance mechanisms, actually the target modifications of ALK just by resistance mutations or amplifications is only a proportion of the mechanisms that the patients can get resistance to therapy. And the bypass pathways appears to be actually a bigger piece of the pie. So that's something also to consider when you are approaching these patients. So with this case, I hope I was able to share with you the resistance of ALK rearrangement can have different multiple mechanisms, and longitudinal tumor sampling may assist in the therapeutic strategies. And unfortunately, even though we have so many advances in this field, we still need more to now overcome the additional problems that we are getting. And with this, I would like to thank my attending Valentina Nardi at Mass General and Alex, who helped me with the case, and Faye, who helped me with the presentation.
Wonderful. Thank you so much for such an informative presentation. It's now my pleasure to welcome Dr. Faye Dong to today's Project Echo video conference. Dr. Dong is an associate pathologist at the Brigham and Women's Hospital, as well as an assistant professor at Harvard Medical School. He'll be sharing his expertise as well. Throughout the presentation, I'd like to encourage everybody to type in their questions and comments along the way. So now I'll turn it over to Dr. Dong to share his slides and begin.
No. Thank you, Kelly, for the invitation to come back, and thank you to the ASCP. It's my pleasure to be here, and thank you everyone for attending. I don't have any disclosures. So in these last few minutes, I just want to share very briefly what we do at Brigham and Women's Hospital, some things we've learned from our NGS program. I'm going to try to show a case of NGS with clinical variant interpretation and kind of reflect on the role or the requirement that we ask from the molecular pathologist in this era of precision medicine. So Brigham Women's Hospital, we are the diagnostic pathology laboratory for Dana-Farber Cancer Institute. It's a tertiary referral center. And so we perform both primary and consultative anatomic pathology, as well as molecular diagnostics. We have a very vibrant molecular pathology laboratory that has 3 different service lines, one focused on solid tumors, one focused on heme testing, and then another one just on NGS, because there's just such a high case volume. So any particular week, we have 3 different molecular attendings that man each of the services. So the core of our service now is this panel called OncoPanel. So it's a pretty standard NGS assay based on hybrid capture of cancer-associated genes. And OncoPanel was first launched in 2013 and has evolved since then. Going from a 275 gene panel to now a 447 gene panel. And that's only the coding regions of these genes. We also capture additional intronic regions of various genes to look for gene fusions. In addition to just changing the chemistry of the panel, we've also changed the reporting. So in 2017, there is a number of guidelines came about that suggest efficacy associated with microsatellite instability and tumor mutational burden. So we now have that in our report, as well as report on a certain subset of mutational signatures. And it's really changed our clinical practice. So before NGS, we did targeted testing, and the selection of tests are based on the pathological diagnosis. So lung cancers would get EGFR and KRAS testing, whereas other cancer types will get other types of targeted testing. Whereas now we really consolidate testing of a lot of different cancer types into the single panel, and that actually improves the efficiency as well as the workflow of the laboratory. We can train our techs to be really good in performing this particular assay. We have a less instrumentation maintenance need because we know that we need to maintain this particular assay for it to work instead of 3 or 5 different instrumentations depending on what we need to do. So there are a lot of challenges for the molecular pathologist now that we have next generation sequencing, something that's so complex. We certainly need to understand the technical basis of the assay, and that includes multiple aspects of the assay, such as specimen evaluation, library preparation, how the sequencing actually works, and as well as the informatics. Each of these steps are pretty complicated and they can generate certain types of artifacts. So if we get a signal that is uncommon, we have to be able to troubleshoot and understand what could have happened that led to this scenario. We also have to do clinical interpretation and give clinical recommendations based on the molecular findings. And that's complex because there are multiple data types that we look at: sequence alterations, such as SNVs, we have copy number variants, we have structural variants. We have to do the technical interpretation to determine whether a signal is true versus an artifact. We have to determine the pathogenicity. Is this particular variant a driver mutation in this patient's tumor, or is it just passenger along for the ride? And also the clinical actionability. So does having this particular finding make the patient eligible for some kind of targeted therapy? So with that, I'm going to jump to one clinical case. And it's a case that someone asked about before. It's particularly interesting. So this is MET intron 13 variant in a case of lung adenocarcinoma. So here is a particular variant, is a 12 base pair deletion. It's seen about 6% of all of our reads. You can see that this particular deletion involves intron 13. It's close to the intron 13 exon 14 junction, but it's not actually covering the junction. So the question is, what does this mean for the patient long-term? So what to do as a molecular pathologist if this comes across your desk? The first and most important thing is don't panic and just try to think logically. And then just think about the interpretable elements. And everyone who's trained around here knows I have 4 questions you always have to ask. So it's pretty easy. What does the gene do? How does that particular variant affect gene function? Is the gene significantly mutated in this tumor type? Therefore, maybe has biological significance in lung cancer or whatever. And is the variant clinically actionable? And we can start to answer some of those questions and start to understand what this could mean for the patient. So we know that MET is a receptor tyrosine kinase and a proto-oncogene. MET mutations are seen in about 3 to 4% of non-small cell lung cancers, specifically exon 14 skipping mutations. And we know that MET exon 14 skipping mutations are associated with response to a targeted MET inhibitor that's FDA approved. So now the question that we have to ask ourselves is, how does this particular variant affect gene function? When you're asking yourself that question, you can take on multiple levels of evidence. So you can say, what has people reported about, this type of MET skipping mutations before? And here's an article from 2016 in which they report on MET exon 14 skipping mutations. You can see that they do have deletions that involve intron 13 alone without involving the intron 13 exon 14 junction. So like, okay, well, maybe this fits or is similar to what we have. Also, you can look at any functional data that might be available. This is the first description of MET exon 14 skipping that was presented in 2006, incredibly. You can see that within that paper, there is a patient with an intronic only deletion involving the 3' aspect of intron 13, 22 base pairs, a little bit larger than ours. But they actually functionally characterized it and demonstrated that this deletion leads to a change in the mRNA product and a change in the protein. So you're like, okay, well, maybe functionally this also makes sense. And finally, I thought this case was really interesting because you have to put this deletion in the context of what we know about cancer biology or how molecular biology works. So if you're thinking of the really key DNA sequences that govern the formation of the splicing complex, you always have a donor site that's not shown here in the 5' of the intron. You have an acceptor site on the 3' of the intron that's right adjacent to the exon, and the sequence is always the same. It's always an AG. There's a branch site near the 3' of the intron that always includes an adenine, and you can see that's the branch site here. And then finally, there's a polypyrimidine tract that's really crucial to the formation of the spliceosome. It's about 5 to 40 base pairs before the 3' end of the intron. It tends to be 15 to 20 base pairs long. And you can see that this particular deletion actually involves directly the polypyrimidine tract. So once you put all of that evidence together, you can say, well, how does this variant affect gene function? You just tell people what you know. Well, this variant is a MET intron 13 deletion involving the polypyrimidine tract near the intron exon junction. Similar deletions have been reported along adenocarcinoma and have been associated with MET exon 14 skipping. Now you've considered all 4 elements, and this is kind of what I consider a complete variant interpretation. So I just want to give you a sense of that. It's something I consider bread and butter molecular diagnostics. Okay, so in summary, when we're thinking of the role and the requirements in molecular pathologists, we have to ensure the quality of reports are generated in the molecular laboratory. And there's more and more that's being asked of the molecular pathologist now, right? So we have to have extensive multidisciplinary knowledge. We have to understand molecular biology and the technical basis of molecular assays. We have to understand anatomic pathology and how disease classification works. We have to understand human genetics and alterations in both health and disease states. And we also have to be kind of armchair medical oncologists, keep track of the current literature and understand current therapeutic options and how genetic results can affect patient care. And then finally, we integrate all of this information to try to provide the best guidance we can for our patients. And that's all I have. With that, I'll end. And thank you for your time.
Wonderful. Thank you so much for such an informative presentation. That was fantastic. So we are now at the top of the hour, and it is time for us to wrap. I'm going to pull up a quick slide here. And we also will be sending instructions for claiming CME and CMLE credit for today's ECHO session. And be sure to save the date for the 2 upcoming ECHO sessions that you see here on the screen. The first one is on November 19th on standardizing specimen handling protocols. And then the second one is on December 2nd on key actionable driver mutations in patients with NSCLC. And so if you want to claim CME and CMLE credit again, please visit the lung cancer page on our website, go directly to ASCP store using the direct links in the chat, and we'll also send you an email for claiming credit to the email address that you registered under. So thank you again, and this concludes today's presentation.